Okay, so good evening everyone. So I am your TA Dipanshi and I'll be taking you problem solving sessions for the course Introduction to Proteogenomics. So I think it's already 6 o'clock, so let's begin with the session. So if you face any difficulties in seeing my presentation or my audio, then please let me know. So your course is Introduction to Proteogenomics, and in this problem solving session we'll be talking about the week 1 topics which you have gone through in different slides which you have obtained for the week 1 and different videos which you have obtained. So in which you have looked into the proteogenomics overview, you have also looked into the introduction to genomics, gene sequencing and mutations, you have studied sequence alignments and a little bit about the transcriptomes. Then you have also heard the lectures which were related to advancement in cancer genomics. So in these sessions, apart from the week 1 topics, we will also be practicing few MCQs. We'll talk a little bit about the current literature which is ongoing, and I'll take your doubts if you have any regarding the week 1 topics. So firstly, I'll be talking about the introduction of proteogenomics, that what does it mean. So proteogenomics is a combined word which is made up of two
Madam, the
words.
voice is
Sorry.
lagging.
Am I audible?
Your voice is lagging, madam.
Voice is lagging. Uh, I guess there might be some network issue. Let me check. So is it clear now?
Yes, now it's fine.
Okay, so proteogenomics, it is a word which is made up of two separate words, which means proteomics and genomics. So it acts as an interface between both proteomics and genomics. So firstly, people used to focus on either proteomics or either genomics. There was no bridge in between. So which misses certain important information which could not be rendered by two of them individually. So then we have to take some bridge between them so that we can combine both the information. So we have more of information and which could also be validated. So for example, if we look into genomics, then we have a vast majority of sequences which we can obtain from the genomics. There could be multiple transcript products which a single gene can exhibit. But to identify whether those gene products are really expressed at the level of protein or not, we also require proteomics. But when we talk about proteomics, which is often studied by using the mass spectrometry, so it misses certain transcripts or certain proteins which could not be captured. So in that case, we cannot say that those transcripts are completely absent. So we also need to have certain information regarding the transcripts, due to which we require the bridge which is proteogenomics. In this, what we does is we have the genomic information, we have the transcriptomics information. Genomics means entire DNA sequence information which we obtain, and transcriptomic means the RNA sequence information which we have, and we can obtain it by RNA-seq or the ribosome profiling. So now we have both these informations. We can convert it into the protein sequence information by customizing it. So we can predict that what protein could it synthesize, and then we can validate it by the proteomics data which we will obtain from the mass spectrometry. So what proteogenomics provide us, it provide us the customized protein sequences which can further be validated with the proteomics data. So in that way we can identify the novel peptides. For example, you have done mass spectrometry and you have obtained certain peptides, because mass spectrometry will give you the peptide information. So now the databases which are the reference protein sequence databases, which are also obtained by the different experiments, they might not have certain proteins listed because the half-life of that protein might be very small, that you were not able to capture it experimentally. So now what help this proteogenomics can provide, now we have the transcript wise information and we have performed its customization, we have synthesized the protein sequence theoretically. So now those peptides could be mapped to theoretical protein sequences. So in that way the identification of novel peptides can be done with the help of this proteogenomics. Then the protein level evidence. So now we have the transcripted so predicted protein sequence from this transcriptomics and genomics data, so it can be validated because we also have the proteomics information. So protein level evidence can be gained with the help of proteogenomics, and we can also refine the gene models. So now how we can refine gene models, because previously certain transcripts which could not be identified, which are now identified with the help of transcriptomics, they might have certain exons which were skipped in some of the transcripts which were experimentally identified, or we can say the proteins So before moving forward, I would like to give you the brief light on the topic of how the gene give rise to multiple transcripts or multiple isoforms. So as you all know that there are about 20 to 25,000 of genes present in human, but the protein diversity is huge. It is about more than 1 lakh proteins. So those genes give rise to such a big diversity of protein by the process of splicing. So splicing is the process where the introns are removed and exons are joined. So other than splicing, the method is the alternative splicing, in which the exons are recombined in different combinations, due to which a single gene can give rise to multiple proteins. So when those combinations are formed, so what can happen is that some exons can get skipped, or there could be some N terminal, C terminal modification of the exons, or intron could be retained. So when we look at the proteomics data which was captured from mass spectrometry, so it could have the proteins which do not have certain exon, or that transcript might have missed. So now with the help of the proteogenomics, those transcripts are now identified. So might be that exon which was previously not listed is now listed. So in the gene models we can label that exon. So in that way proteogenomics will also help us to refine the gene models. So these are some of the purposes that why proteogenomics was established, but now on its applications are huge. So we will look into that one by one. So these are the proteogenomics shared resources. So in proteogenomics we integrate the RNA-seq and a mass spec based proteomics, and this data is generally used to identify the variants and mechanisms of their functional regulation. So we can also predict that what kind of function that particular protein would perform. So in this, the high resolution mass spectrometry could be performed at the level of proteomics, and next generation sequencing for the RNA-seq data is performed. So these two are combined along with the customized bioinformatic programs which are given by the Galaxy project. So Galaxy project is a kind of project for those people who don't have very much information about the coding, so they can also use this pipeline to analyze their data, that what kind of data they have obtained after RNA sequencing. So these are some of the resources, and with the help of proteogenomics you can also go for the metaproteomics. So not just the single genome, you can also look into the genome of microbes which are present inside us along with our human genome. So in that way you can study the host microbiome interactions. So in that way different applications of proteogenomics can be studied, and there are multiple resources through which you can go through this information. So there are certain papers which are related to proteogenomics. So one such mini review is given by the Kelly Ruggles, whose lecture also I think you have gone through. So in this paper they have talked about the current perspective in proteogenomics, and this was published in 2017. So in this they are telling about that how different samples of the macromolecules, the DNA, RNA and proteins are taken, and what kind of functionalities can be performed on them, what kind of information can be obtained. So if we have the DNA sequences, we can go for whole genome sequencing, in which the entire genome could be sequenced, or we can also undergo whole exome sequencing. So whole genome is like you are going to sequence everything, the coding sequences as well as non coding sequences or repeats, everything. But when we talk about the whole exome sequencing, then you will be focusing only on the exonic sequences. So in that way you will only be looking at the coding regions within the genome. So in this way it is kind of cheap and quick as compared to whole genome sequencing. And then if we talk about the RNA sample, so if we have RNA sample, we can go for the RNA sequencing, we can sequence the RNA. And if we have the protein sequence, then we can perform mass spectrometry to get the fragments, and then those fragments are further converted to the peptide sequences, and those peptides are further looked into the reference database to check that from which protein that peptide could have come. So in that way the peptide is labeled with the protein information. So when we have so now let's move to the DNA and RNA information. So now we have performed sequencing, we have the reads, or we can say the short fragments which we have. So now those fragments can be mapped to the reference sequence. For example, we have a human genome, and now we have taken the sample from from a person who is suffering from cancer. So we have obtained his DNA. Now we will do sequencing, and we'll compare it with the reference human genome, that where the changes are coming, which genes are disturbed, where are the mutations. For that we will perform assembly. So assembly is the way when we can map those small reads onto a bigger fragment, so that we can combine those reads and construct a single read or single genome sequence. So in that way the alignment methods can be two way. So one is de novo read assembly, in which we do not have any reference information. We will simply place the reads and we will look for the overlapping regions so that we can construct a single long read. Whereas if we have a reference information, then it's quite easy. We will simply align the reads to that reference and we'll see that which sequences are present. So now after performing the alignment and after doing the assembly, we will construct a single scaffold sequence. Now we will check for the variants, that where are the variations coming from. Either there are single nucleotide variations, or there are some indels or gene fusions, or there are splice junctions. So these all things could be checked in those sequences. And then customized sequence databases is constructed, because these are going to provide us with the DNA sequences would be converted to protein sequences. So those conversions will be done with the help of the codon table which we have. So we will be predicting the protein sequence here. But in mass spec, we already have the fragments, the peptide sequences, which we are comparing with the already available reference protein sequences. So it is a validated protein sequence, whereas in the case of DNA and RNA sequencing, we have the protein sequence which is predicted. So now we will compare both of them. We will validate that the protein sequences which we have obtained, whether they are present in the reference protein sequences or not. So after that, what we can do is we can do genome annotations. We can map peptides to the genome, or we can perform mutation analysis. We can check that with the normal genome and the cancer genome, how they vary, where are the variations. We can also do metaproteomics, in which we will study that to which species that genome or peptides are belonging, because in human various microbiome is also present inside us. So when we obtain those samples, so it will also help us to map those peptides to different species, or to study the metabiome which is present inside us. And then there is another review in which they have talked about the solid tumors, that how we can identify the neoantigens. So the neoantigens are the proteins which are expressed in the tumors. So other means when the normal proteins get mutated and expressed in tumors, so those are referred to as neoantigens. And it is easy to target the neoantigens because they are different from the self proteins. So in that way, if we target those neoantigens, then the autoimmune disorders could be prevented. So this paper is about that, and you can go through this paper. In this also they have talked about the similar procedure, that how the data can be collected, and at last you will be getting the protein sequences, so you can do biomarker discovery, you could predict the disease and novel pathways. So these things are done when you have the cancer sequence, and you will compare it with the normal human genome sequence, and you will predict that where are the things are changing. So in that way you can predict that whether the disease will occur or not. So what are the features which are linked to that disease, and pathways can also be explored. So in genomics, we have looked into the two terms, that are whole genome sequencing and whole exome sequencing. But other than these, there is one target sequencing also present, so in which you will be focusing on the targeted sequences. So in that case, for example, I already know that I am studying the breast cancer and I want to emphasize or focus only on 2-3 genes. So in that case, instead of sequencing my whole genome, I'll sequence the targeted regions and compare their sequence with the reference sequence and check that where are the mutations or SNVs are present. So for that, target read sequencing could be done. So when we talk about whole genome sequencing, so it covers all the genes as well as known coding DNA. Whereas in whole exome sequencing, you will be covering only the exome, which are the exonic sequences. And in case of TRS, you will be covering the targeted regions or targeted sequences, which could be either genes, or which could be any region which you have selected. So the accuracy associated with whole genome sequencing is quite low, because you are focusing on the entire genome, whereas in whole exome sequencing, the accuracy is good. And in case of targeted sequencing, accuracy is quite high, because you are focusing on a very smaller region as compared to these two. And the variants which you can study with the help of whole genome sequencing are the SNVs, indels, and structural variations. And in case of whole exome sequencing, you can look into the coding SNVs, the variations which are coming due to the in the coding region, indels and copy number variations. Whereas in TRS also you could look for the SNVs and indels. So whole genome sequencing is quite expensive, because you are going to sequence whole genome, and its depth is about 30x. Whereas in case of whole exome sequencing, it is cost effective, and its depth is 50 to 100 reads. Whereas in case of TRS, it is most cost effective because you're only focusing on the targeted regions, so it's quite cheap, and the depth is 500x. That is 500 reads you can get in a single run. So now we will be talking about the methods of sequencing. So till now, is it clear? Should I go slow, or so you can also let me know, because this is my first session, so I don't know if you're not understanding anything. Then you can let me know. I'll reduce my pace. So
Yes, it's clear now. Like till now, it's clear, ma'am.
okay, so now I will be telling you about different sequencing methods. So there are you have heard about the next generation sequencing, but before moving to next generation sequencing, so the sequencing is being categorized into three main categories. So one is first generation sequencing, then comes second generation and third generation. So most of the people, they refer to as the sequencing techniques after second generation as next generation sequencing. So those first generation and second generation, they are the short read sequencing, but now as we have advanced a lot, so we can also go for the long read sequencing. So let's begin with the first generation sequencing. This involves Sanger sequencing, Maxam-Gilbert, Sanger chain termination sequencing. So these primitive sequencing techniques could only sequence the smaller fragments. So this first generation could only sequence from 500 to 1000 base pair fragments. Whereas if we move to second generation sequencing, which involves the 454, which is the pyro sequencing, so Lexa, Ion Torrent, and Illumina. So they are the high throughput sequencing methods which could sequence 50 to 500 base pair fragments, and the accuracy has improved in these Illumina sequencing as compared to the Sanger method. And first generation sequencing also faced the problem, the limitation, because the Maxam-Gilbert method involved the chemicals which are carcinogenic or harmful chemicals, and in Sanger sequencing also it was the chain termination method. So due to which these methods were quite of discontinued, and we have focused on second generation methods. Then means primitively the third generation methods have also come up, which are PEGBio and Oxford Nanopore, which can sequence the native DNA in real time with single molecule resolution. So their resolution has also improved, and the sequencing could be of tens of kbs of fragments on average. So how you can see how long sequence reads it could sequence at a single run. So due to which third generation sequencing are more favored. But if we look at the level of accuracy, then the next generation sequencing or the second generation sequencing is better as compared to third generation sequencing. But if we want to do it quick, then third generation sequencing is favored. It depends on your desirable output or what you need and in what time. So on that way you can select one of the sequencing methods. So this is the comparison table in which I have compared all the NGS methods along with the first generation and the third generation. So means from now on the second and third generation will be called as next generation sequencing only for the simplicity. So these are the next generation sequencing methods, and the Sanger one is the first generation. So you can go through this table if you want to know more in detail that what are the characteristics of these different sequencing methods. So now I'll give you the brief introduction of all those sequencing methods, that what is basically done. So in the Sanger sequencing, as we know that it is the dideoxy chain termination method. So in this what happens is that when you have the fragment which you want to sequence, so what you will do, you will provide it with the different polymerase DNTPs primer, as well as you will also provide the DDNTPs. So the application or the principle behind this sequencing is that if you will provide different DDNTPs, so DDNTPs are the dideoxy NTPs which lack the OH group at their 2' position. So sorry, at the 3' end it will lack the OH group, due to which it will not be able to extend the DNA chain. So in that way it will terminate the chain. So here you can see we have a DNA fragment which we want to sequence. Firstly we require a primer so that we can allow the DNA synthesis. So primer is present, polymerase will now start the synthesis of the nucleotide on this leading strand. So it will take the DNTPs, it will like to bind it. Now we also have DDNTPs. So at some point of time it will take the DDNTP which will be the complementary to the parent or the template strand. As soon as this DDNTP will bind to this template strand, it will stop the elongation of the DNA chain, and hence this particular daughter strand which is synthesized, it will get terminated, because DDNTP will lack the 3' OH group which is responsible for the extension. So this is the principle behind the Sanger sequencing, that whenever this uh, DNA or the daughter strand will be terminated, so those DDNTPs are the fluorescently labeled molecules. So they are labeled with some fluorescent signal, and each of these DDNTPs will be labeled individually with the separate fluorescent tags. So whenever the termination will happen, it will release some kind of fluorescent signal which could be captured, and that device will capture it and it will give some peak. So with the help of those peaks you can identify the sequence which is present. So in that way the sequencing is done with the help of Sanger sequencing. Now let's move to the next sequencing method, which is in the next generation sequencing method, Illumina method, and which involves the MiSeq, HiSeq 2500, HiSeq 3000. These are the different variants of the Illumina method. So what is simply done in this case, you have a DNA sequence and you will bind the adapters. So firstly the DNA sequence is converted to single stranded form, and after single stranded form we will fragment it because it is a long sequence, so we have to fragment it, and then after fragmentation we will attach the adapters on both the ends of those fragments which we have done from the genomic DNA. So now sequence library is prepared. So first step is the library preparation. So in this way library is prepared. Now we have to generate the clusters. So cluster generation means that for a single fragment you have to generate multiple clusters, so that means you can increase the depth of your sequencing with the help of the cluster generation. So now you have attached the adapters to this, and in this case you use the flow cell. So flow cell has some kind of oligos which are attached to the flow cell and which are complementary to these adapters. So whenever you will provide this fragments along with the adapters to the flow cell, so those adapters will bind to these oligos present on the flow cell.
Madam,
Hello,
uh,
yes.
s- sorry to interrupt. Sorry to interrupt, but would you please repeat that how we are creating the library and then we are forming the clusters?
Yeah. So what we does is that we have a genomic DNA, we will convert it into the single strands, and then after the single stranded DNA is obtained we will fragment it. So we will fragment into smaller pieces, and after doing the fragmentation of the entire genomic DNA we will bind it with the adapters on both the ends. So the ligation is done, so which is known as the adapter ligation. And there are different methods for the ligation of the adapters, because when you will fragment, so it is made sure that the blunt ends are obtained, and the adapters are ligated on both the ends of the fragmented DNA. And now when the adapters are ligated on the fragments, so second step is the cluster generation, in which we have a flow cell. So in Illumina sequencing we have the machine contains the flow cell. So flow cell is a surface in which there are oligos attached to it. So those oligos are the oligonucleotide sequences which are complementary to the adapter sequences. So when you will send this DNA sequence or the sample of the DNA fragments to the flow cell, then the adapters will bind to these oligos. So in this case the blue ones which you see here, these are the oligo sequences. So adapters will bind to those oligos, and then a straight DNA fragment will be present. So after that there will be a bridge amplification cycle. In bridge amplification what it will do is that we will provide it with the polymerase and it will synthesize. So this method is a sequencing by synthesis method in which the fragment will be synthesized. So now this single stranded DNA will convert into double stranded DNA, because we will provide the DNTPs as well as the polymerase along with this. So different means the prerequisites or the necessary things which are required for the replication, they will be provided. Now double stranded DNA will be synthesized on the surface of flow cell. So as you know that these oligos will be complementary to these adapters, and these adapters are so now when the double stranded DNA will be synthesized, the topmost end, so as soon as double stranded DNA synthesis, one of the strand will be removed and the other strand will again form a bridge. So what it will do is that from the 5 prime end it will fold and it will attach to the adapter. So in that case a gate or a bridge like a structure will be formed. So similarly again the polymerase will come and DNA synthesis will begin. So in that way multiple such bridges will be formed, so cluster will be formed. So that means a single DNA fragment will be converted into multiple clusters. So multiple similar sequences will be generated in this case. And when the multiple sequences will be generated, so when enough of the clusters are generated, then you will again start with the their replication, in which you will provide the DNTPs which are labeled with some kind of fluorescent signal, so that now you can capture when the sequencing step will start, then you will be able to know that which of the nucleotide is binding, so that you can capture that signal and you will be able to sequence. So ultimately in the sequencing you are going to provide those labeled nucleotides, and it will give multiple reads, so multiple clusters are there and multiple reads will be obtained simultaneously in the flow cell and those will be recorded. Now the fourth step is the alignment and data analysis. So as we have seen that multiple fragments were present, so we will obtain multiple reads and those reads could be overlapping, so we will try to uh, align it to the reference genome and we will be able to sequence the DNA which we have provided. So in that way the sequencing is done in this way in the Illumina method. So is it clear? If you want to read it more in more detail, then you can go through certain YouTube videos. So I can also give you the link of those videos in the end of my presentation. So because by looking at those videos it will be more clear. Um, Means if you look at the text, then it becomes a little difficult and challenging to make you understand, but if you look at the videos that how those things are happening, so it will be quite easier for you to understand. So this is a overview that how the Illumina sequencing works. So should I proceed? Do you have any doubt?
Yes, yes, I understood. Yes.
So yeah, so in this case the pair end, you have observed that there is a paired end sequencing because the adapters are linked to both the ends. So paired end sequencing is better as compared to the single read sequencing, because in the pair end you will be attaching the probes at both the ends, or the adapters at both the ends, so you will be able to read it through both the directions. So that's why the pair end sequencing is beneficial as compared to the single end, because in this case you can sequence it from the 5' to 3' end as well as from the 3' to 5' end. So that is one of the advantage of the paired end sequencing, and it would also help you to detect the genomic rearrangements or the repetitive sequence elements, and you can also look that whether there are some gene fusions, or you can also identify the novel transcripts with the help of paired end sequencing. And in this slide I have provided you with the comparison between the paired end and single end. So for more detail you can go through the slide. I'll not go into the detail because it is not related to the course. It means because we don't have to go through the detail of this, so you can go through in your free time if you want to read it in more detail. So next is the ion torrent sequencing. So ion torrent sequencing is based on the principle of the ionic exchange, that how the change in pH will means what is the change in pH will tell you the which of the nucleotide is present. So in this way you will be able to sequence the nucleotides, because each nucleotide will change the pH by some different extent or some unique extent, due to which you will be able to know that now which nucleotide is attached. So this method is also sequencing by synthesis method, in which you have a DNA fragment and now you are synthesizing the complementary fragment of the daughter strand, so you will be able to know that which of the nucleotides are binding, and whenever a new nucleotide will come it will lead to change in the pH, and in this way you will be able to capture that which nucleotide has bind by the looking at the peak. So I will not go into much of the mechanism of this method, but this is the overview that how it detects the sequences. Okay, so the next so in this case I have provided you with the detail that step by step that how the ion torrent sequencing works. If anybody is interested, then you can go through the slide. And then comes the third generation sequencing methods, which involves the Pacific Biosciences sequencing in nanopores. So firstly we'll talk about the Pacific Biosciences sequencing. So these methods are the long read methods, and it is also known as single molecule real time DNA sequencing technologies, because in a single time or in the real time you are sequencing, you don't have to fragment the genome, because in the previous methods you have to fragment your genome into smaller fragments, and then for each fragment you identify the sequence and then you have to align or assemble it. So whenever you do assembly, so there are the chances that there could be errors, so that's why those methods were having some error. But when we talk about the third generation sequencing method, as they are the single molecule real time sequencing methods, you are directly sequencing the entire genome, so you don't have to fragment it into smaller regions. That means you don't have to assemble the reads in the end, so in that way the error is reduced to some extent. So in Pacific Biosciences also you have a high quality double DNA fragment which is present. Now in this case they attach the single molecule real time Bell libraries. So these are the fragments which are attached to the end of the DNA fragments, and these are circularized. So what it will do is that it will help us to circularize the entire DNA fragment, and whenever this DNA fragment is circularized, now there are nanovels. So in the Pacific Biosciences you will observe that the slide will have the nanovels which will be having the polymerase enzyme, and whenever you will give the DNA sample which is attached with the adaptase, so it will start its synthesis. So this is also a sequencing by synthesis method in which the DNA is provided. Now it will try to synthesize the daughter strand, and it will have the DNTPs which are fluorescently labeled. So whenever it attaches some complementary nucleotide, it will give some signal, the fluorescent signal, because when DNTP will bind, it will release that fluorophore and the signal will be captured, and its intensity will be captured, that which of the nucleotide is now binding. So in this way the sequencing can be done in the real time for the entire genome or the entire DNA sequence. So in this way this is the Pacific Biosciences sequencing. And then comes so this is the detailed step by step things which are involved in Pacific Biosciences. So you can go through this if you want to know more in detail about the PEG biosequencing. So then comes the Oxford nanopore sequencing. So in this case, in the previous method we have talked about the nanovels, but in this case we have the nanopores, and they read the DNA sequences in the real time. So for example, here you can see there is a membrane which below which there will be a solution or electrolyte will be present inside the membrane, and there is a nanopore which is present on the surface of membrane. And what you will do is you have a DNA sample, you will bind it with the adapters which will allow it to attach to the motor protein, and the motor protein will allow your DNA to pass through this nanopore. So this passing will happen because you will give some current. So whenever the current will be flowing, so it will allow the double stranded DNA to pass through this motor protein, and this motor protein will guide it through the nanopore. So whenever that double stranded DNA will be passing through the nanopore, then below the membrane, as I have told, that there will be a solution which will be electrically charged. So now the ions will be because there will be charge flowing through that, so there will be change in the charge, there will be deflection, because whenever the nucleotide will pass through that nanopore, so there will be deflection in the charge, and those deflections will be captured in the form of peaks, and ultimately the charge deflection by each individual nucleotide will be different. So you will be able to capture that which nucleotide is present at which position. So in this case you are not synthesizing some other complementary nucleotide, but you are just checking the deflection or the change in the charge. So this is the Oxford nanopore sequencing, and it is also a long read sequencing method in which you can see there is a min ion which is one of the device used for the Oxford nanopore sequencing, and this is means as small as which can be held in hand. So in this way, with the advancement of sequencing methods, the sizes of the devices have also reduced, and the read length or the accuracy has increased to some extent. So these are all about the sequencing methods. And in this case, if I have to generalize the sequencing method, so what are the four major steps which we do in sequencing method is, firstly we extract the DNA, because DNA is the first step, because we require the DNA which we want to sequence. So first step is the DNA extraction. Second step is the library preparation, because we require the library because we have to give some fragments which we want to sequence, because before the third generation sequencing methods have arrived, previous methods they were kind of sequencing the short reads, so we have to fragment the longer sequences into the smaller fragments. So in that case we have to prepare the library in which we have to break down into smaller fragments and attach the adapters. Whereas if we talk about third generation sequencing, so in library preparation we simply attach the adapters to it in case of the Pacific Biosciences, so which are known as smart bell. And then sequencing steps comes in which will capture the either change in charge or change in the hydrogen positive ions or the fluorophore signal. So there are different things which are captured and which are ultimately helping us to sequence the DNA fragment. So now the last step in the sequencing is the analysis, because now we have obtained those data, and now how to analyze that data. So now we will look at how do we analyze the sequencing data, because we have obtained the DNA sequence. Now how to proceed further? So first thing which is involved is the FASTQ file, because after sequencing you will be obtaining the FASTQ file, which is the FASTA quality file. So the sequences, DNA sequences or the RNA or the protein sequences, so these sequences are present in the FASTA format. So FASTA format is a kind of a file format in which the sequences are saved in the computer. So in case of FASTQ, which is the FASTA quality file, so it will tell you the quality of each read. So not just the sequence information, it will also tell you the quality. And where does this quality come from? Because each sequencing method is having different methods to detect the sequence. For example, the fluorophore signals or the change in the current. So in that way different peaks are obtained in every sequencing methods. So those peaks, in all the cases it's not always that the peak will be so clear that you will be able to distinguish that this is the particular nucleotide. In some cases those peaks could not be that strong to distinguish between different nucleotides. So in that case, we will get the poor quality reads. So in this FASTQ file, it will provide you with the information of the sequence along with its quality, that what is the quality, whether the peak was so sharp to confidently say that this particular nucleotide is present in the read, or whether the quality is poor. So when you will look into the FASTQ file, so firstly it will give you the sequence name, that what is the name of the sequence, because if you have the multiple fragments, so in that case each fragment will have different sequence name and its length will also be given. So this is the sequence information. Then comes the DNA sequence. For example, if I am doing DNA sequencing, then it will be giving me DNA sequence. After that there will be the quality line break, which will tell me that now I am going to give the quality scores for my DNA sequence. So these quality scores will be given in some ASCII character form. So here I have given the different ASCII characters which are used to give the scores to these sequencing methods. So for example, here we see BBF. So what does these mean? So in this, if you see the uh, P error, so this is the quality. So quality is from 0 to as long as more than 40, so it can go till 100, the quality can go till 100. So 0 means the quality is 0, that means it's a poor quality, you are not able to uh, for example, in this case there is no poor quality, but for example if I see a exclamation mark, which means that the C, so the signal is so poor that I am not able to distinguish between all the 4 nucleotides, but I think that it is more nearer to the C, so I am labeling it as C. So in that case the quality will be 0, which means that signal was not so accurate to distinguish. And as we move forward, the score, the quality is increasing. So increased quality means sharper peaks were obtained, so the predictions or the sequencing which is done, which it will be more accurate. And then P error, so P error, actually the scoring is done with the help of Fred. So in Fred, the error means the probability that the particular nucleotide which is obtained at a particular position, what is the probability that it could be an incorrect base call. So whenever we do sequencing, whenever we label the sequence, we call it as a base calling, that which base is present at this position. So whenever there is a probability that it could be a faulty base, so which is referred, which is given by the P error. So if the P error is lower, so P error will low towards the right side, whereas quality will improve towards the right side. So if you see more of the alphabets, then it means the quality of your FASTA file or the quality of sequencing is better. And if you see these special characters or the numbers, which means the quality is quietly low. So this is all about the FASTQ file and how to interpret the FASTQ files. And we will prefer that the quality scores should be greater than 30, so that the probability of error is as low as 0.001, that means 1 in a 1000 base pair. So this is about FASTQ. Then comes the BAM file. So after obtaining the read and after getting the quality scores, now you are sure that this sequencing is done correctly. Now you have to perform its alignment to the reference sequence, you to check that where of means from which region this sequence is coming from. So that is done by the alignment method, and the alignment will give you the output in the form of BAM file, which is the binary alignment map, and it is a compressed version of SAM. So SAM is also the sequence alignment map file, which will give you the entire information about the alignment. So in this, what you does is you provide the reference means reference sequence, and the reads will be aligned to that reference sequence and the information will be given to you. So this is the output of the BAM file, how it appears. So first is the header section, which will tell you the information about your reads, that which of the sequence is which of the sequence is this and what is the reference. So this is the header section. Then comes the alignment section, which will tell you the alignment, where that particular read is aligning to, which will involve the query name. So this is the query name, and then the flag, which will indicate the alignment information, that whether it is paired or aligned. So it will give information about the read. Then comes the reference sequence name. For example, its chromosome or the transcript ID will be given by this third field in this. Then there will be position, map Q, which will tell you about the mapping quality. Then the cigar, which is the summary of alignment, that whether there are some insertions, deletions, or matches, mismatches. Because in this you are aligning it, you are aligning the sequence with the reference. So now this file is giving you the information that where are the sequences are exactly matching, or if there are some mismatches, then that information will also be given you by the cigar. Then R next will give you the reference sequence name of the primary alignment of the next read. Then P next, T length, so which will give you the number of bases which are covered by the reads from the same fragment. Then the sequence information will be given, its quality will be given, that whether this read quality, so if information is available then it will tell you about that, if not available then star will be given. And there are some optional fields. If you have some more information, then in the optional fields you will find it. So this is all about the BAM file, and in your course I don't think you have to go through the detail of these files. I'm just giving you the quite idea that what does it mean, what does these files contain. So this is all about the BAM file, and its ultimate aim is to align your sequence to the reference, to know that this read is coming from which of the position in the genome, or which fragment, either it is coming from certain gene, certain region of the gene, or intersection of the gene, or intergenic region. So all information can be given you with the help of BAM and SAM file after alignment. Now third thing is the variant call format, because our ultimate aim of sequencing is to understand that whether there are some variations present in our sequencing read or the DNA fragment which we are sequencing from the normal or the reference genome. So variant call format is for that. So this format will tell you that in your query sequence where are the changes. So this will tell you about, so firstly there is a VCF header. Header will tell you all the details of your reads. Then comes the body. So body will tell you the chromosomal positions, and in the chromosome, so for example there is a chromosome 1. So each chromosome have multiple, so the DNA will be arranged in the form of multiple positions. So this will tell you which position, so in reference what is the sequence at this position, and alternate will tell you that whether that nucleotide fragments have changed to some other sequence or not. So in this way it will be telling you about whether if there are some deletions, or there are some single nucleotide polymorphisms, or whether there are large structural variations due to deletions. So this file is ultimately going to tell you that what are the regions where the variation is happening when you are comparing your file. So in BAM you are able to compare, you are going to align it, and then the variant call format will come after the alignment, that where are those alignment varying between the reference and the query sequence. So query sequence could be anything, either you have taken the DNA sample from some patient who is suffering with cancer or some other disorder, and not just the, if you want to compare different species, then also you can undergo this sequencing method. You can compare that where two sequences are changing. For example, I have two bacterial species and I want to check that these two encode the same gene, but this bacteria is growing in these conditions and this bacteria is growing in some other conditions. So in that way I can compare that gene sequence, and I can do variant call format. I can perform that analysis, and I can get the file in the variant call format to compare that where the variations are coming from. So this is one of the format. So now this format is actually capturing the mutations or the changes which are coming. So mutations is the one of the region which is leading to these changes. So mutation is a mechanism in which there is change in the DNA sequence of the cell. So whenever there is change in DNA sequence, that is known as mutations. So one thing you should emphasize on is the DNA sequence, because there are many things which are also happening. There are some epigenetic modifications which are also happening, so that are not referred to as mutations. For example, if some cytosine is kind of getting methylated, so we will not call it as a mutation. if it is changing the promoter binding or anything. Mutation will only be referred to as the DNA sequence change. And mutations may also be caused by the mistakes during cell divisions, or if there is some problem in our replication machinery, or if we add some DNA damaging agents, or if we are exposed to certain agents or the chemical reagents which could affect our DNA. So in that case the mutations can be observed, and these mutations are not always harmful, they could be beneficial also, and they could also have no effect. There could be some neutral mutations which are going on with evolution. So if these mutations are occurring in the exosperms, then these mutations will be heritable. Whereas if these mutations are occurring in some other cell of the body or the somatic mutations, so these will not be inherited. And here are different ways of classifying the mutations. On the basis of origin, they could be spontaneous or induced. So spontaneous are the mutations which are occurring in the absence of any known mutagen, which are spontaneous due to errors in the replication machinery or other reasons. And induced are the mutations which we are inducing. We are going to induce those mutations either through external mutagen agents, or we are exposed to UV light. So in that way the mutations which are arising due to that reason is the induced mutations. Then on the basis of cell types, mutations could be somatic or germline, which I have discussed. And then effect on the function. So now mutations could affect the protein functionality in different ways. So it could lead to loss of function, which are also known as knockdown or null mutations. So it could lead to the elimination of the normal function. It could be hypomorphic, which means it could lead to the reduction in the normal functionality. It could be hypermorphic, it could either activate the genes in such an extent that it could be expressing at a higher extent, so it will increase the normal function. And there could be gain of function mutations also, so which are expressed at incorrect time or inappropriate cells. Whenever those mutations, whenever that function is not required, then also it has gained the function. So that is also one of the outcome of the mutation. And on the basis of molecular change, these mutations can be categorized as transitions. So when we talk about the DNA sequence, so in transition means that a purine is converting to another purine. For example, when A was present, it is replaced by G, and pyrimidine is replaced by another pyrimidine. So thymine is changed with cytosine. So in that way these are transitions. Then comes transversions, when the purine is changed with the pyrimidine and vice versa. So transversions will have drastic effect as compared to transitions, because in transitions they are similar, the nature is similar, the purines to purines, so it won't have that drastic effect as compared to transversions. Then comes insertions. So if one or more nucleotides are inserted, so that is referred to as insertion. Then deletions, if one or more nucleotides are deleted, which is referred to as deletion. And then comes effect on translation. So on the basis of effect on translation, the mutations can be categorized as silent mutations. So silent mutations also referred to as synonymous, which means that there are changes in the DNA sequence, but that change is not visible at the protein sequence, because we know that in the codon table there are certain triplet codons which codes for the similar amino acids. So in that way, if the change is occurring at the third position, so or some other position, then it will not be affecting us, our amino acid sequence which is being encoded. So that kind of mutation is referred to as synonymous mutations. Means the mutations only occurring at the nucleotide level, but it could not be read at the protein level because amino acid is same which is encoded. Then come missense mutations, or the non-synonymous ones, which means that the nucleotide is changing and it is ultimately also changing the amino acid sequence. So you will also be able to observe it at the protein level. So these are non-synonymous mutations. Then comes nonsense or termination mutations. These are the mutations in which the nucleotide is changing in such a way that now it is coding for a stop codon. So it will lead to premature termination of our protein, because now the codon will be converted to stop codon, so no amino acid will bind to that and it will terminate our protein. So these are nonsense mutations. Then comes frame shift mutations. So frame shifts will shift the frame, reading frame. So mutation, for example, some deletion or insertion happens, so it will change the reading frame, due to which amino acid content will also get changed. So these are frame shift mutations. So this is all about the mutations. Now we will talk about the SNPs, because our main aim was to study the SNPs. So SNPs are the single nucleotide polymorphisms in which there is only a single base point which is changing in the DNA. So here you can see this is the DNA and this is the common genotype. So in the reference AT is present, whereas if a variant genotype, for example, you are looking at the normal person and the person who is suffering from cancer, and you are comparing its gene. So you might see that there is a variant genotype which is leading to altered protein. So in this case, previously AT was present, but in this case GC are present. So that single nucleotide has changed, and now if that single nucleotide has changed, so till after that, whenever it will go for the replication, the that particular mutation or particular change will go on increasing. So in this way, this is the SNP site, and in this way we compare the single nucleotide polymorphism or single nucleotide variations among two genes, which we will see that after doing the alignment, we will look into the SNV and we obtain the variant call format files to check that what are the mutations linked to particular disease, so that in the future we will be able to predict that this person might suffer from this type of cancer, because previously we know that this person is suffering from this cancer. We have obtained his DNA sequence, we have mapped it to the reference sequence, and we know that the mutations are coming at these genes at these regions. Now we will take the genome of any person randomly and we will check that what are the sequences present in his genome, and then we will be able to predict that whether he will suffer from the same disease or not. So these are the helps which are provided with the help of these methods. So these things can be done, we can check. And in this case I just want to add on that the altered protein, it is not always that in the cancer only you will, or in the disease state only you will observe altered protein, but normally also you can observe the altered protein, because in different persons, due to different epigenetic conditions or different environments they are exposed to, they could produce altered proteins. So same the genotype means the genotype could have undergone some change and alter protein is synthesized, which might not be detrimental to that person, which might be beneficial, or means which could have the variant effect among both of them. So it is not always related to disease or harmful phenotype or harmful outcome. Yeah, so in this I have provided you the databases which will give you the information about the single nucleotide variations. So you can go through them. I have also provided the URLs if you want to go into the detail that what kind of information is available. So DBSNP, ClinVar, 1000 Genome Project, Exome Aggregation Consortium, Genome Aggregation Database, and Cosmic, which you have gone through in your lecture, which is the catalog of somatic mutations in cancer, then OpenSNP and GoExome. So these all provide you some kind of information other than means including SNP, they will also give you the other informations of the variants as well. So you can go through them if you want. Yeah, so now when you check these variants, so you must be aware of the sequence alignment thing, because how you will be able to capture these variants, for that you have to perform the sequence alignment. So sequence alignment will tell you that the two sequences, how similar they are or how different they are. So there are two terms with which students get often confused too. One is sequence homology and other is sequence similarity. So when we say sequence homology means that these two sequences, they share some common ancestor. These might have originated from the same ancestor. For example, if we talk about the human, we have certain genes which are also present in the mouse. So which means that we might have some common ancestor from where those two genes are coming, and we both share some kind of genes. So we can compare those sequences, and that is referred to as sequence homology, which means that two sequences, they have some kind of evolutionary relationship. And when you compare two sequences, so its output could be yes or no. That means either two sequences could be homologous or either they could not be homologous. But when we talk about the sequence similarity, which means we are comparing the similarity between two sequences, we are aligning them and we are checking that how similar they are. So sequence similarity will be in the range, which will tell you that how much similarity between two sequences is present. So it will be a range, and if sequence similarity exceeds certain threshold, then those two sequences could be referred to as the homologous sequences. So sequence homology could be derived after comparing the sequence similarity. But if sequence similarity is poor, then we could not refer them to as the homologous sequences. And what is the hypothesis, or what we have observed so far, is that if two sequences, they share the higher sequence similarity, then they could also have the functional associations. That means they could perform the similar functions, and they could also have the similar structure. So AlphaFold is one of the advanced tool for the structure prediction which has come up. So it is also based on that same principle, that if two sequences have a similar, if two sequences have similarity then they could also have the structural similarity as well. So in that also the principle is same. You will perform the MSA, which is the multiple sequence alignment. You have unknown sequence, you will try to align it with the multiple sequences, and you will see, you will predict its structure, because you will check that if it's having the high sequence similarity to particular sequence whose structure is already known, then you can predict the structure for the current or the sequence in the question. So this is about the sequence similarity. And then there is another term which is also confusing a lot, is sequence identity. So many students think that the sequence similarity and sequence identity are the same things, but no, they are different. When we say sequence identity, we mean that if we are comparing two sequences, then those two nucleotides, if we talk about the DNA sequences, and amino acids, if we talk about the protein sequences, so those two, if they are identical, then only we will say it is then only we will calculate the sequence identity. But when we talk about sequence similarity, so in that case um, there are some matrices which calculate that if one nucleotide is exchanged with another nucleotide, what is the effect on that particular DNA sequence. So in that way it has some kind of scores which are provided to it. So I'll give you the example of a protein sequence. So in case of sequence identity, if my protein sequence have proline, and let's assume it is now replaced with the lysine. So in case of sequence identity, those proline, when it is mapped with the aligned with the lysine, so I will give it a score 0 because it is not identical. But in case of sequence similarity, when a particular amino acid is changed with another amino acid, we will see that whether they have some kind of similarity between them. So amino acids are of different types, some are charged, some are non-polar, some are polar. So if polar is replaced with the polar amino acid, then it will have less influence on the protein. So in that case it will have some kind of score. So in case of sequence similarity, we will also take into consideration that. And if we talk about DNA, then in case of transitions and transversions, so transversions will have adverse effect as compared to transitions. So sequence similarity will also be different. And in sequence identity, we will be make sure that the sequence should be identical, it should be complete match. So that is percent identity. And in similarity, you are little bit relaxed that whether it is not identical but it is sharing some kind of features, then also you will have some score. So there is a range of scores which are present in the sequence similarity, and there are some matrices. So I won't go into the detail of those matrices, but they exist, due to which you will be able to calculate the sequence similarity. So in identity it will be 0 to 100, but when you see sequence similarity, it will not be in the range 0 to 100, its range could be anything. So this is one thing. And sequence homology is already discussed, its output will be yes or no. And one rule of thumb is that if two sequences are more than 100 amino acid long or 100 nucleotide long, they are considered to be homologs if 25% of the amino acids are identical. So yeah, I told you that to call two sequence as homologous sequence, we will check that particular threshold of sequence similarity should be met. So this is the 25% threshold, and it could change depending that which sequences you are comparing, but this is general thing which people consider. And here I am telling you a little bit about the two algorithms of the sequence alignment. So sequence alignment could be local or global. When we say local alignment, so local alignment means that you are looking for the local regions which are similar among two sequences. You are not caring about the entire sequence. Whenever you are comparing two sequences, you are only focusing on certain regions which are aligning in both of them. So that is local alignment. When we say global alignment, you are kind of making sure that the entire sequence is aligned to one another. So here you can see that this sequence is aligning to this particular fragment, and you're not caring about the terminal regions. So this is local alignment. You are just focusing only on the local region which is aligning with the better score. But in this global alignment, you are forcing your alignment to align it end to end. So end to end alignment is the global alignment method, and the algorithms which are involved in local alignment is Smith-Waterman, and in global alignment the algorithm which is involved is Needleman-Wunsch algorithm. And then the another thing is pairwise sequence alignment and multiple sequence alignment. So as the name suggests, pairwise sequence alignment, that means you are aligning a pair of sequence, only two sequences are considered. But when we say multiple sequence alignment, which means you have a single sequence and you are mapping it to multiple sequences. For example, you have the unknown sequence, you got the genome. For example, you're working with the microbes, you got some unknown microbe and you want to sequence its genome. You don't know to which species it belong to. So now you will kind of do multiple sequence alignment to check that what could be the possible species it could belong to. So that is one of the case of multiple sequence alignment, in which you are mapping one single query sequence to multiple sequences at the same time, which is multiple sequence alignment. And in this I have provided you different tools which are generally used. So in case of pairwise sequence alignment, you have heard about the BLAST. So BLAST is majorly used for the uh, pairwise sequence alignment. So the method is means its method is pairwise sequence alignment. Then emboss, needle, emboss, water, K-tuple and Kamer algorithms. Whereas for the multiple sequence alignment, the tools which you use is muscle, maft, clustal omega or clustal w, and T coffee, K-align, and these tools are available at the ensemble site. So sorry, EMBL site. So in this I am also providing you with the link if you want to explore these tools. So you can go through these links whenever I'll share the presentation with you. So is it clear or should I just revise it again? Is it clear? Can somebody confirm?
Just I'm following.
Okay. Yeah, so now let's move forward. So now I'll give you the brief idea that how the BLAST works. So BLAST is a basic local alignment search tool. So previously I've told you about the local alignment method and the global alignment method. So local is just trying to match to the best region in the two sequences. So BLAST is based on the local alignment. So BLAST will, this is the server page of the BLAST. So it will provide you with the nucleotide BLAST options, in which if you have a nucleotide query and you want to map it to the nucleotide sequence only, then you will perform the nucleotide BLAST. Then comes BLAST X, which means you have translated nucleotide information and you are going to search it with the protein sequence information. So you know that there are 6 frames of translation which could exist. So you have, after sequencing the DNA or RNA, you are kind of predicting what could be the protein sequence it could generate, or you are synthesizing a customized library, which we have seen in the when we were discussing proteogenomics. Then we know that after sequencing we generate the customized protein sequences. So those are generated using these, that is referred to as translated nucleotides. You are translating them. You don't know that whether it is the experimental protein sequence or not, but you are translating it. So for example, you have those translated nucleotides and you want to know that which protein it is. So you will do BLAST X, which takes the translated nucleotides and search it against the protein sequence database, and it will tell you which protein it belongs to. Then comes T-BLAST N, which means now you have the protein information and you want to check which of the translated nucleotide database it is coming from. So in this you would search, the query will be protein sequence and the database which you are searching it against is translated nucleotide. Then comes protein blast, which means you have a protein sequence and you are blasting it against the protein sequence database. So this is protein blast. And then there are means these things are elaborated in this region. So this is about the blast window. Now I will give you the example so that you will get more detail about blast. So in this case I have taken the BTB domain 9. So this is a gene. So I have obtained this sequence information from NCBI genes just to perform a little exercise. So you can simply go to NCBI gene and you can search any gene because you want to blast it and you want to find that whether blast is performing the alignment in the right way or not. So in this I have taken this gene and I have selected some of the region of this gene. I am not selecting the entire sequence, I am just selecting this particular region of the gene. Now this is the nucleotide sequence. So what I have to select is BLAST N because I want to search that nucleotide sequence against the nucleotide database. So I have selected BLAST N, I have entered my query, I have simply pasted it. If you want to means upload your file, you don't want to paste it, then you can simply download the FASTA information from the NCBI gene and you can simply upload your file from here. You can choose your file and you can upload it here. You can also give the job title if you want, because in some cases there are sequences are very large that it is not getting blasted early. So in that case you can give job title and you can make sure that the output or the result is further given to your email address. So you can also give your email address in this, so that you get it through email. The results will be provided to you. Now the second thing what you have to do is, after giving the query, is to select the database which you want to choose. So in this, by default they have selected the standard database, which is non-redundant. Then there are some other databases which are also present, but this was by default, so I am not changing it because I don't have any specific requirements. Then in organisms, you can select organism of your choice. For example, if you know that you're searching it, you want to search it for the Homo sapiens, then you can select Homo sapiens. And if you don't know that which organism it is coming from, then you can leave it blank. It is optional. So in this, I know this is a human gene, so I've selected organism to be Homo sapiens. And then there are certain other options, so you don't have to go through these options. Now in the last, you have to select the program. You can optimize it for the highly similar sequences, which is megablast. And there are some other also, that more dissimilar sequence or somewhat similar sequence. So it depends, because in my case this gene is already listed, so I know that I want the highly similar sequence. But in your case, might be your query is variable and you don't know that whether it will be highly similar to some already existing sequence or not. So in that case you can change it, but this is by default setting, so I'm not going to change it. Now I will simply click on blast, and now after some time, it will take some time to run this job, and at last it will give me output in this form. So it is giving me the job title, and these are the program which I've used, the database, the query ID, everything it will give, the length of the query. So the query, the DNA sequence which I have selected, which was of 980 nucleotide length, so that is also given. And this is the kind of description of the result which it is producing. So firstly it will tell me that that particular nucleotide sequence could come from these sequences. So these information are already available in the BLAST database. Then it will tell me the organism from which it is coming. So in this case Homo sapiens is given. Then it is telling me about the score, that what is the alignment score. So after alignment there will be some score which is generated. So what is the total score? Then query cover. So query coverage, it is kind of the coverage, which means that if your query sequence is certain amino acid, for example in this case, as it is a nucleotide query, so it is 980 nucleotide long. So how much of these nucleotide is aligning to that particular sequence or the reference sequence which is present in this database. So in this case it is giving 100%, which means that all these 980 nucleotides are aligning to 980 nucleotides in the reference sequence as well. Then comes e-value. So e-value is also very important when you judge the BLAST results, because e-value is the expect value, which will tell you that what is the probability of this alignment generating by chance. So it should be as low as possible, because it should be as low. So if it is lowest, then it is the best BLAST hit or best result which you have to select. Then comes percent identity. So in this case it is calculating the identity between two sequences. 100% means each nucleotide is identical. So in the max score and total score, it is telling you the percent similarity. But in case of percent identity, it is telling you about the identical matches which is present between the query and the reference sequence in the BLAST. Then the accession length, and then accession number. So accession length is the sequence which is present in the BLAST. What is the length of that sequence? And this is accession number. If you want to go into the detail of that sequence, you can click on this link and you can check. So this is the BLAST result, and you can also explore the graphic summary, alignments, taxonomy if you want to go into more detail. So in this case, I have gone through the alignment thing. So in alignment, you will be getting the result that if you have a query, so at which position it is mapping to the reference sequence, and what are the nucleotides which are present. So in this case it is showing 100% sequence identity, but if the sequence identity is not 100%, then this alignment will help you to check that where the sequences are changing. So in this way you perform BLAST. So I hope it is clear, because this BLAST thing is also present in your assignment. So in this way you can do BLAST and you will get the results. Now I have also given the sequence alignment tools which are used for the multiple sequence alignment, such as Cluster Omega, Emboss. So in BLAST it was the pairwise sequence alignment. One sequence is aligned to one sequence at a time, but in this you can provide multiple sequences at the same time and you can compare. For example, if I have 5 different genes present in human, and I want to check that whether their promoters remain the same, what are the promoter regions. so that things can be compared. I can, in the query, I can give multiple protein sequences and I can compare all those sequences between one another. So in BLAST you are giving one sequence, comparing it with the entire database of BLAST, but in multiple sequence alignment what you are doing is you are giving your own multiple sequences and comparing among themselves. So that is multiple sequence alignment, and these are the different tools which you can use. They have certain advantages and disadvantages, so you can select them depending on your need which you want. Now the last, not last but the second last topic of week 1 was the transcriptomics. So when we say transcriptomics, it means the entire RNA information which is present in your cell at a particular point of time, that is transcriptomics. And why do we need to study transcriptomics? Because I've told you that the single gene can give rise to multiple transcripts which can synthesize multiple proteins. So there are certain transcripts which are exhibited in certain states. For example, there could be certain transcript which is only observed in the cancer state and not in the normal state. So in that way, if you know which transcript is expressed in cancer state, so that particular transcript you could target it and you could design some drug against that particular transcript. You can check the regions which are causing that variation, because those transcripts, they are generated by different combination of exons and different other mechanisms such as exon skipping or intron retention or enter 5 enter CTEL modification of exon. So you can check that where the modification is coming. So that's why the transcriptomic is helping us to do that. So in this diagram you can see that in vivo there is a DNA and we are only focusing on a single gene in the genome. So after transcription it will synthesize the pre-mRNA. So pre-mRNA will undergo splicing, in which introns will be removed and exon will be combined. Then mature mRNA will be synthesized, which will move from the nucleus towards cytoplasm so that it could convert to protein. So now this is in vivo thing which is going on. Now in vitro what we can do is that after obtaining this RNA sequence, we have to fragment it because we want to sequence that RNA. So in transcriptomics we will perform RNA sequencing. So we have to fragment those mature mRNA into smaller fragments. And now when we do sequencing, so we do not go for the direct RNA sequencing, we have to convert it into cDNA. So for that we have to perform reverse transcription, which will convert this RNA sequence to the cDNA sequence. So the difference between the DNA and the cDNA is that cDNA is the copy DNA and this is the DNA which will lack any known coding regions. So if you compare the DNA sequence, it will be composed of exons, introns both. But when we talk about cDNA, so this DNA is synthesized from the mature RNA, mature mRNA. So it will not have any intronic regions, it will be the coding region, or it will be the exomic DNA sequence. So now cDNA fragments will be obtained, you will perform high throughput sequencing and then sequences will be obtained. So any of the technique could be used to perform this RNA sequencing. And then in silico you have to perform the sequence processing, the alignment, the BAM files will be generated, and then you will be able to identify the splice variants. So in this way they are telling you that there are two splice variants which are observed, splice variant A and B, and the difference is that in splice variant V, one of the exon is skipped. So in this way transcriptomics help you to identify the splice variants which are present in your sample. So this is about the transcriptomics and the units. So now if you are doing RNA sequencing, then there are multiple reads which are generated. So there are some units which are associated with these reads, that how you will calculate the number of reads and how better your RNA sequencing output is. So for that we have different units. One is FPKM, which is the fragments per kilobase of transcripts per million mapped reads. So this is generally used for the pair end RNA-seq. And if we talk about RPKM, which is the reads per kilobase of the transcripts per million mapped reads, so it is for the single end RNA sequencing. So paired end and single end, which we have discussed earlier, that when you are sequencing from both the ends, that is paired end sequencing, and when you are only sequencing from the one end, then that is single end sequencing. So FPKM, it is the normalized expression unit, and it will take into account both the transcript length as well as total number of mapped reads in the sample. So it will represent the number of fragments which are mapped to a transcript per kilobase of the transcript length. So it will tell that how many fragments, for example you are considering this much of the transcript length, so how many fragments are mapping to this particular length, And if the more and more fragments are mapping, that means you will be more confident in establishing that particular fragment, in sequencing that particular region. So in that way, this is the FPKM. This will tell you that if the more and more fragments are mapping to certain length of the transcript, the better is the RNA sequencing. Then comes RPKM, in which we will only count the total reads in the sample and divide it by the 1 million to obtain the RPKM score. And this normalize for the sequencing depth and give you the reads per million. So it will not take into consideration the fragments, it will only consider the reads. And then comes TPM, which is the transcript per million, and this is the expression unit for the gene or transcript quantification, when you are just looking into the number of transcripts which have been quantified in the RNA-seq data. So this is TPM, and this has the normalized read counts for the transcript length and total mapped reads while accounting for the presence of alternative isoforms and multi-mapping reads. So in RNA-seq, this will tell you that how many of the transcripts have been identified which are alternate transcripts or alternate isoforms. And the only difference between RPKM and FPKM is that FPKM is used for the paired end RNA-seq, and I think there is some of the question related to this in your assignment, so this might help you to solve the assignment. Now let's look into the formula. So if you want to go into the detail that how these scores are calculated, then you can go through the slide in which I have provided you. Okay, my screen is not visible. Can someone confirm? Is my
It
screen
is
visible?
visible.
Okay, somebody is
Visible, ma'am.
okay, somebody is saying that my screen is not shared. Okay, I think you should again connect if my screen is not visible to you. Okay, so this slide I have given the formula that how these scores are calculated, so you can go through that. Okay, so now I will be talking about the cancer genomics, because the main hype which is created of the proteogenomics is through its application in the cancer genomics or the cancer study. So in cancer genomics, what we do is we will be studying the totality of the DNA sequence and how the gene expression is changing between the tumor cells and the normal cells. So cancer genomics help us to identify those genes and to identify that what are the genes responsible to particular cancer and what are the pathways which are influenced, what are the mutations which are coming. So this all things when we study, it is referred to as cancer genomics. And with the advancement of techniques, with the advancement of sequencing techniques, alignment techniques and proteogenomics, now we are able to study the cancer in a better way. So before 2012, these are the different methods which were present. So first was DBSNPs. So previously what people used to do, they used to align and they used to look for the SNPs, that where are the nucleotides are changing, what are the variations coming. So with those variations, people try to predict that if these variations are present, then this is the fate of that gene, this would lead to a cancer state or not. So these were the previous things. Then slowly and slowly different sequencing techniques were launched. Cosmic came up, which tells you about the somatic mutations in cancer. TCGA came up. So there are different projects which got established, which were collecting more and more information, because with the more and more information in our hand, we could analyze it and our predictions will become better with more and more data in our hand. So these are different projects which came up till 2012, and after that there is a huge rise in the techniques, and now it has improved, the cancer predictions have improved. So there are different you can say repositories which are present, which contains the information of the cancer. So one is Genomic Data Commons and NCI, National Cancer Institute. So National Cancer Institute has taken the initiative to collect more and more data from, it has collaborated with different agencies, different laboratories, different people, so that it could gain more and more data for the public welfare. So anybody could use that data and they could help to design the drugs regarding cancer or precision medicine. So Genomic Data Common and NCI, so they are the data repository, they allow to store the data, and you can also analyze the data from their site. You can get any data regarding the cancer genes. Then comes CPTAC, which is the Clinical Proteomic Tumor Analysis Consortium. So these all are the databases, you can say, which contains the information related to cancer. So CPTAC is aimed at advancing the cancer research. It is also integrating the large scale proteome and genome analysis. So now data is present, analysis is present, different places analysis is present. So these are doing collaboration. These are kind of combining all those information at this single place so that it could be better to analyze. And then there is Apollo Network, which is the collaboration among the National Cancer Institute, Department of Defense, and Department of Veterans Affairs. So they have come hand in hand to integrate the proteogenomics into the patient care so that the personalized medicines could be designed and the better healthcare facilities could be come up in terms of the cancer treatment. So these all are the advancements which have happened through these years in the cancer genomics. So I am giving you the quite overview of the GDC portal. So this is the Genomic Data Commons portal. When you go through this portal, I have also given you the link if you want to explore it. So in this, it is telling you different projects which they have performed on different patients, what are the primary sites from which the data has been obtained, how many cases they have studied, how many files they are providing you, how many genes which are observed in these cancers, and how many mutations. So all these informations can be obtained from this GDC data portal. So it is a repository, and in this they are also telling you about the different tissues or different primary sites where the cancer genes are mostly observed, mostly expressed. So these are the cases, which are the tissues which are the cancers which are mostly observed. So each and every information is present in these portals. If somebody wants to study, then they can simply go to these portals and download the data and then do their study. So this is the advantage of this GDC portal. Then the CPTAC, which is the Clinical Proteomic Tumor Analysis Consortium. So it is also a data portal and it is taking information from different sources. So TCGA and other portals, it is combining the information at a single place, and anybody could they could either download the information, they are also providing you with the antibody informations, assay portals. So these all portals are present in the single CPTAC server. So it is also a kind of a repository where you can go and check for the data which is present, and they are also providing you with the help of analyzing that data as well. So you can also analyze that cancer specific data. And then these are the objective of CPTEC. So the overall objective was just to identify those proteins that derive from the alterations in the cancer genomes and their related biological processes. So these all informations are present in the CPTAG. So you can check that what are the information they are providing. And the three main components of the CPTAG is Proteome Characterization Center and Proteogenomic Translational Research Centers and Proteogenomic Data Analysis Centers. So these are the three components which combine and which further analyze the data and give you the information. So these are the main three components. Then the Apollo network. So this is also the network coming from the Office of Cancer Clinical Proteomics Research, and it has different tiers, and this Apollo stands for the Applied Proteogenomics Organizational Learning and Outcomes. So this network is a collaboration between NCI, Department of Defense, and Department of Veteran Affairs, and it incorporates the proteogenomics into the patient care. So in this, they are also combining the proteogenomic information, and it is same as CPTAC only. Under CPTAC only, there is Apollo network. So in this, they are combining the information, and these departments have come together, they have collaborated, and they are trying to design better healthcare things. And ultimately there is a cancer moonshot program which was initiated by the government in America. So now they are joining hand with the other countries so that the cancer treatment could be designed, because till now the cancer is the disease for which we do not have better drugs, or means we could not cure it, means we do not have that much drugs known. There are certain cancers which do not be cured, although we have some therapies, but they are not 100% successful. So we need to have some more therapeutics related to this. That's why this moonshot program came up. And now the NCI is so NCI has started this cancer moonshot, and now they are joining hands with other countries such as Australia, Canada, Germany, Taiwan, Japan, Sweden, and Republic of Korea, so that these all countries will come together, and if more people will collaborate, then it will be easier to them to design the personalized medicine and to study in a better way. So till now they have done more than 2000 publications, more than 49 clinical trials have been performed, more than 30 patents are filed. So such programs are very much needed so that better healthcare could be established. So this is in terms of cancer. So similarly, the other disease, for other disease also, these kind of collaborations would be helpful. So these are some of the advancements, and now I will be talking about few MCQs. So we still have 35 minutes. So in this we will be solving few MCQs, and you can unmute and answer these questions if you have any idea, and then we will see the answer and we will discuss that why this is the answer. So the question number 1 is that Sneha is a microbiologist who has a keen interest in studying environmental microbiome. For her research project, she wants to study the genome of a fungus, Cladosporium, which is found in the salt marsh sediments. Which next generation sequencing platform will be most suited for her project? So these are some kind of sequencing platforms. One is Illumina MySeq, one is mass spectrometry, one is Oxford Nanopore MinION, and one is PegBio. So she is dealing with the genome of Cladosporium, which is a fungi. So now which of the sequencing technique do you think would be most suited for her project? And I'll give you a little hint. If you have gone through the lectures, then I think Kelly has talked about this. So anybody
Yeah,
wants to?
actually, she told in the lecture that we should use Illumina MySeq for the microbiology uses.
Yeah.
A, ma'am.
Okay. Anybody else wants to answer? If they think other than Illumina MySeq, some other technique should be used. Okay, now let's see. Yeah, so its correct answer is Illumina MySeq, because we know that fungus Cladosporium, so its sequence is not that large that we should look for the Oxford or PegBio. So Illumina MySeq is sufficient to sequence this genome, and then option B was mass spectrometry. So this is for the protein sequence. Uh, this will give you the protein sequence information. So this is not the this would not be the correct answer, and Oxford Nanopore and PegBio is also not the correct answer. So Illumina MySeq is the correct answer. Okay, so now let's look at the next question. Yeah, so the question is, DNA sequencing is the process of determining the order of nucleotides in the DNA. Which one is the correct order of sequencing reaction? So which of the option do you think is the correct order? Option A is library preparation, sequencing, pooling, demultiplexing, and alignment. Option B is library preparation, pooling, sequencing, demultiplexing, and alignment. Library preparation, alignment, pooling, sequencing, demultiplexing. Or option D, library preparation, pooling, alignment, demultiplexing, and sequencing.
Option B.
Okay, so anybody
B,
else?
ma'am.
Okay, okay, someone else wants to answer? Okay, now let's see. So the correct answer is option B, which is the library preparation, pooling, sequencing, demultiplexing, and alignment. So now let's look at the option A, why it is not correct. So what you do is, pooling is a kind of a clustering step. So when we were talking about the uh, Illumina sequencing, we were talking that after the library preparation we go for the clustering, so which is also the pooling. You were increasing the number of fragments for better sequencing. So pooling is the second step, then comes sequencing. After sequencing you go for the demultiplexing. So till now we have not discussed about demultiplexing, but demultiplexing comes in when we are talking about multiple samples, because multiple fragments are present. So in some cases we have multiple samples which we are present. So in multiple samples we provide different labels to each sample, so which is done in the form of barcodes. So in that case we have to demultiplex it to sort that which fragment that particular sequence belong to, and then ultimately we perform alignment to know that to which region that sequence belongs to. So option B is the correct. Okay, now let's move to the third question. Sorry. uh, so the third question is, single nucleotide polymorphism is a germline substitution of a single nucleotide at a specific position in the genome. So in the below figure it represents different types of SNPs. So you have to identify the SNPs in each case, A, B, and C. So the first thing represents the no mutation. So this is the normal DNA sequence. This is the normal RNA sequence, normal protein which is synthesized. Now in A, B, C there are some changes, and changes at each level. So you have to tell what kind of event is undergoing in these three stages. So option A is that in A synonymous mutation is observed, in B nonsense, in C missense, Similarly, in option B, A is nonsense, B is synonymous, C is missense. In C, A is synonymous, B is missense, C is nonsense. And in D, A is missense, B is synonymous, and C is nonsense. So we have already discussed about mutations. So I hope you will be able to answer this. Okay, what do you
Yes,
think?
option D.
Which option?
Option D,
D.
because the C one you can see that it's a stop codon, so from
Okay.
that we can say it's nonsense. And yeah,
Yeah. Anybody else wants to answer? Okay, so now let's see what is the correct answer. So yeah, correct answer is option D. So now why option D? Because as Srijita said that this C is the stop codon. So if stop codon comes, it is known as the nonsense mutation. And then in case of B also you can see that if there is no mutation, tyrosine is present, and in case of B also tyrosine is present. So I have told you that the synonymous mutations are the ones which are only detected at the nucleotide level, but they could not be detected at the protein level because the change in the nucleotide is such that it is not visible at the amino acid level. Same amino acid is being encoded. So in this case, it is a synonymous mutation. If you look at the one, it is cysteine, and previously in the reference it was tyrosine, so which means there is change in the amino acid. So whenever there is change, it is known as missense mutation, or you can also call it as the non-synonymous mutations. So option D will be the correct answer. Now let's move to next question. So to remove the variations of different genes, normalization serves as a crucial pre-processing step. Which one of the following statements is not correct regarding the normalization of RNA sequencing data? Okay, so these are few options. Option A says that FPKM and RPKM both are expression units for normalized RNA sequencing data. RPKM is usually used single end RNA-seq, whereas FPKM is used for paired end RNA-seq. Option C says FPKM is usually used for the single end RNA-seq, whereas RPKM is used for the paired end RNA-seq. And option D says the only difference between the RPKM and FPKM is that FPKM takes into account that two reads can map to one fragment. So now in this you have to tell which of the statement is not correct. So we have previously discussed about these three units for the RNA-seq reads. So these are the normalized units. So what do you think is the not correct option?
So option C.
Okay. Anybody else wants to answer? Yeah, so let's see. So option C is the correct, because we have discussed that FPKM is used for the paired end RNA-seq, whereas RPKM is used for the single end. So this is not correct, whereas option A is also correct. Both are the normalized, both are the expression units for normalized RNA-seq data, and RPKM is used for single end, FPKM for the paired end. This is also correct. And similarly, the main difference between both of them is that FPKM takes into account that two reads can map to a one fragment. So in that way, it will not count it as two, it will count it as one. Whereas in RPKM, it will count it as two separate reads. So this is also one point which you can keep in mind. Okay, so now let's move to next question. So now you have to select the correct option. So what are the major components of CP-TAG coordinates? So we have just discussed it. So option A is proteome characterization, option B is genome characterization, option C is metabolite characterization, option D is proteogenomic translational research, and option E is proteogenomic data analysis. So I'll give you a little hint. So it has three major components. Now you have to tell me that which of these options is correct. In this ABCD represent these things. So what do you think are the major components of CP-TAG coordinates?
Second one, A, B, and D.
A, B, D, ma'am.
Okay. Okay, so anyone else wants to answer? Okay, now let's see. So the correct answer is option number 3, which is A, D, and E. So in CP-TAG, it is about proteome characterization, proteogenomic translational research, and proteogenomic data analysis. And it is not linked with the genome characterization, it is emphasizing on the protein characterization. So proteome, proteogenomic translational research, and proteogenomic data analysis. So these are the three main components of CPTAC coordinates. Okay, so let's look at the next question. What is the role of NCI, which is National Cancer Institute's Genomic Data Commons, which is GDC? So option A is data generation, option B is data collection, option C is data mining, option D is data repository, option E is data sharing. So these are the options. What do you think is the role of NCI GDC? So we have looked this also.
D, ma'am. Only D.
Only D, data sharing. Sorry, data repository. Okay. Anyone else wants to answer?
D and E.
Okay, D and E, data repository, data sharing.
BDE.
Okay, data collection, data repository, data sharing. Okay. Okay, so let's see. Oh, sorry. So the correct answer is data repository and data sharing. So in NCI, Genomic Data Common, what it does is it is a data repository and a data sharing. So this is not going to generate any data, and data collection is kind of a misleading option here. But it is a repository which is storing the data and which is sharing the data. So it is not going to collect it, but it is already having the data and which is being uploaded to it. So yeah, the data collection is kind of misleading here, but the correct option is data repository and data sharing, which already contains the data which is present in it, and then it will share the data, and it is not going to collect the data. So you're not it is not, means if you have the data, then it is, you're not able to give that data to it. It is not collecting it. It already have a repository which has the data stored into it, and that data could be shared. Okay, so now let's move to the next question. Sorry again. Yeah, so which of the following groups works with the collaboration of Apollo network? So we have also discussed this as well. Food and Drug Administration, Department of Veterans Affairs, Department of Defense, and National Cancer Institute. So which are the groups have collaborated for the Apollo? So these are the options.
B, C, D.
B, C, D. Okay, V, A, D, D. Yeah. So anyone else? This is very simple. Okay, so I will tell you. Yeah, so BCD is the correct option, because Food and Drug Administration is not part of Apollo network. But one more thing which I want to tell you, that when we talk about the Cancer Moonshot program, so in that case, if you look at the different agencies which have collaborated, so in that case FDA is present. But if you talk about Apollo network, then FDA is not present. Then Department of Veteran Affairs, Department of Defense, and NCI has the collaboration in the Apollo network. So let's look at the next question. Now in this case, you have to match these databases with the correct options. So the first database is the Cosmic, second is DBSNP, third is GoExome, and fourth is OpenSNP, and these are the descriptions. So you have to tell to which description these databases belong to, and if you don't have idea about these, then means you can simply if you know a single answer, then also you'll be able to answer it. It's quite straightforward. Okay.
D.
D. Okay, and anyone else wants to answer? Okay, now let's see. So the correct answer is option D. So why option D? If you don't know about these three, then means till now you have known this Cosmic. So Cosmic is the catalog of somatic mutations in cancer. So here the correct option for first will be the database of somatic mutation in human cancer, which means one will be matching to third option. So in this case, option D is only having the one matching to 3. So simply if you don't have the information about 2, 3, 4, then also you will be able to answer it. So now let's look what the DBSNP provides you. So DBSNP is linked to fourth, which means full collection of identified SNPs. So it is a database of SNP which will provide you the full collection of those SNPs which have been identified. Whereas option 3, which is the GoExome, so this is a SNP database from lung, heart and blood disordered patients. So this is the GoExome, and then comes OpenSNP. OpenSNP allows you to upload the SNP data. So this is an open database in which you can upload your data. So these are the databases and their descriptions. I hope it is clear. Yeah, so these are the questions. This is it. Now I will talk about the current literature or the current resources which you can go through related to cancer, and this have been published recently. So it is a resource and which says the pan-cancer proteogenomics expands the landscape of therapeutic targets. So in this, proteogenomics is kind of helping you to understand and identify more and more therapeutic targets related to the cancer. So when we say pan-cancer, which means multiple cancers they are studying. So the key takeaways in this resource were that currently there is limited drug target information with FDA. So FDA provides you with the approved drug targets. So which will also tell you the drugs which have been approved. So they are fewer than 200 proteins. So now there is need to design more and more therapeutic targets. So in this paper what they have talked about is the comprehensive proteogenomic analysis, in which they have integrated the CPTAC data from 1043 patients, and they have also considered additional data sets with which they have identified 2863 druggable proteins and their dependencies, aiding accurate drug target predictions. So in this they are telling that how the proteogenomics has helped to expand the landscape of therapeutic targets, because from 200 proteins, means not just 200, less than 200 proteins, they are moved to 2863. So which tells you that how much strength the proteogenomic analysis has. And now you can also identify the new targets. So new target identification strategies. So by combining the proteogenomics data with the genetic screens, it revealed that targeting the tumor suppressive gene loss and prioritizing the mutant KRAS peptides provide a detailed landscape for drug development. And this is a link. So this is the database where you can go and you can identify that which are the new targets which have been identified, and the information is provided in this server. So this picture or this flow chart is given in this resource, which tells you that which are the resources from where the information have been gathered. So for the genomic resources, the information is gathered from GTEC and TCGA. So GTEC is a gene tissue expression database which gives you the expression of the genes and the transcripts in the normal tissue, that in the normal state which genes are expressed. And then TCGA is the cancer genome atlas, which tells you that in cancer which genes are expressed and what is the sequence, where are the variations. So TCGA is also a bigger platform like GDC data common. This is also having the information gathered from multiple resources. It contains different projects, different patients and different samples, and then analysis have been performed to check that where are the mutations coming from, SNVs, copy number variations, all those information are present in TCGA. So which is related to the cancer specific expression. So these genomic resources information is taken. Then cell line screens. So deep map is used, and in which knockdown and drug response have been checked. So this information is also obtained from the deep map. Then CPTAC, which is used for taking the tumor and normal tissue from 10 cancer type information. Then drug targets have been obtained from drug bank, and then other resources which provides the information of drug targets. And then proteomic resources which will tell you about the information related to protein which is present, such as cancer atlas and pep query. So they have protein based information, whereas the GTEC and TCGA they have genomic based information. So all these resources have been combined, and then their analysis has been performed to identify the different biomarkers. So in this way, what can be done with these resources is expression and activation driven protein dependencies, in which you can compare in normal and tumor state what are the cell lines, what is happening at the level of functions. So these things can be done at the expression level when you are going to express those genes or proteins. And then KRAS mutant peptides as putative public neoantigens. So the neoantigens, as I have mentioned earlier also, it is a term in which a particular protein is synthesized or is formed in the cancer state and which is not present in the normal state. Those are referred to as neoantigens. And the KRAS gene is the gene which is mostly observed in different cancers. so it gets mutated, so due to which if you screen for this KRAS you will be able to predict better that which cancer is, so whether the cancer state is present or not, whether it is normal state or the cancer state. So these KRAS mutant peptides could be searched. Then synthetic lethal partners of tumor suppressor loss could be identified, and identification and confirmation of tumor associated antigens. So in this way, in this paper, these all things have been discussed, so which ultimately gives you the idea that how important these proteogenomic resources are for the better personalized medicine and therapeutic design for the cancers. And then there is another um, short review article which you can go through, which provides you the noble insights into the cancer treatment. So other than proteogenomic treatments, there are many other treatments which have also been proposed. So you all are aware of the chemotherapy which goes for the cancer patients, but other than that there are several other techniques which are coming up, such as bacteriotherapy, so in which people say that if we inject a person with a certain bacteria, then they will release certain proteins which will help us to target the cancer tissues in a better way. So bacteriotherapy has also come up, then microRNA therapy, then targeting ribosomes, then high peg. So these are some of the cancer therapies which are advanced therapies which are coming up, and means uh, so these techniques are not that much accurate till now, but still we need some more time to further advance in them. And then cancer drug delivery systems have also come up, aptamer, polymeric nanoparticles, so how to deliver the drugs. So these are some of the normal so advanced techniques which have come up. So the key takeaways from this review is that, so bacterial therapy in cancer treatment, so in this they are talking about the bacteria. For example, the FDA approved BCG strain for the bladder cancer, it exhibits the anti-tumor properties and can deliver the therapeutic agents like cytokines and antibodies directly to tumors. So it is a kind of a delivery, so drug delivery thing. So you can give that drug through the BCG strain, and that particular bacteria will go to particular tissue and release that cytokines and antibodies to the tumors to stop that tumor, and it will act as an anti-tumor agent. So this bacteria will stimulate the immune responses and can influence the metastasis through biofilm formation. Then the second is targeted therapies and gene editing. So advanced cancer treatments such as microRNA based therapies. So what you can do with the microRNA, you could, if you know the particular gene and particular, for example, particular gene is responsible for cancer, so you can uh, kind of silence that gene. You could knock out, or you could do microRNA silencing based thing to regulate its expression. You could target the ribosomes, or CRISPR-Cas9 gene editing could be performed so that you could means regulate the expression of that protein. You could stop it or you could increase it depending on what kind of gene it is. For example, if it is tumor suppressor gene which suppresses the tumor, so you want it to overexpress so that it could control the tumor. For example, tumor suppressor genes are involved in apoptosis, and in cancer you see there is a means increase in the cell growth at particular site. So to inhibit that cell growth you need apoptosis to kill those cells. So in that case tumor suppressor genes will help you, so those genes could be their expression could be increased. On the other hand, the oncogenes which are promoting the cancer, so those oncogenes could be suppressed using these CRISPR-Cas9 gene editing methods. So these are certain methods which could be used to deal with the cancer. Then innovative drug delivery systems. So other than the targeting, the drug delivery system has also advanced. So nano vaccines and targeted drug delivery systems have come. Okay, so sorry, there was some network issue with me. So Yeah, so I was talking about the review article that other than the targeted therapies and gene editing, the innovative drug delivery systems have come up in the recent time, such as nano vaccines or targeted drug delivery systems, which includes the aptamers and nanoparticles. So means this is all the advancements which have been observed in the cancer treatments. Okay, so this is all about my presentation. If you have any doubts regarding your assignment or the week 1 topics, then you can ask me. I'm open for doubts and questions. Or if there is any doubt regarding the today's topics, then you can let me know so I can go through it again. And if you want some other reading material or anything, means any kind of help, then you can let me know, so I will add those things also in my end of my presentation, so that when you access the presentation you will be able to get those information as well. So there is no doubt. Okay. Yeah, I will be sharing these presentations. So there is I think they have shared you with this spreadsheet in which I'll be uploading the daily presentations, sorry, not daily, but the weekly presentations, and I will also be providing you the YouTube links of these recorded sessions. So due to the network issue, I hope the recording has not gone bad. From the next time I'll be try to set in the better network area so that there is no issue. So anything else, because we still have 8 minutes. Yeah, you will be getting in your mail public sheet, they will be sharing you, and if you don't get by the next time, then you have to let me know. I'll share it in this also. Yeah, so in that public sheet I will be giving you with the folder or the Google Drive link in which you can access these presentations, as well as the YouTube link from where you can go through the online live lectures if somebody have missed, or if somebody have some doubt to certain regions, so they can go through the video again. Okay, thank you so much. Thank you everyone for patience and for answering. It is motivating for me. Okay, so I don't want to waste your time. So in this time, let's talk about certain things. Uh, okay, so other than cancer, uh, I'll discuss with you a concept. So in proteogenomics, so I will tell you that how the proteogenomics came into the picture. So firstly, long back when human genome was not sequenced, so at that time betting used to happen, that people used to bet that how many genes would be present in human. So everybody was guessing that human genes would be in lakhs, because the protein variability which we observe, it is in lakh. So when the human genome was sequenced, then everybody was surprised, because genes are only 20 to 25,000, whereas proteins were up to lakhs. So then they checked that why this difference is present at the gene expression level and the protein expression level. So when they were checking, they observed that there are different processes which are undergoing. So one such thing is the alternative splicing, and not just alternative splicing which brings about this proteome diversity, but other things are also there, which are alternate translation initiation and alternate translation terminations. So as you know, AUG is the start codon. So when we say alternate translation initiation, so translation is protein synthesis. From mRNA, when protein is synthesized, that is referred to as translation. So the translation begins from the start codon, which is AUG. So in some cases what happens is RNA has multiple AUGs. So that means it could start its transcription from translation from multiple sites. So due to which there is alternate translation initiation. So these are known as alternate translation initiation sites, and these could also lead to different proteins that could be synthesized. So one reason is alternate translation initiation. Second reason is the alternative splicing. So alternative splicing means when introns are removed and exons are combined. So in some cases the spliceosomal factor, which is responsible for this splicing, sometimes it's not able to recognize the splice sites. So the splice sites are the sites which are, means you can also call them as signals, which are present on the introns, which are kind of flags which are telling the spliceosome that come here and cut us. So sometimes spliceosome is not able to recognize those signals. So in such cases, that exon which is attached to the intron, that could get skipped, or in other cases the intron could get retained along with the exon. So intron retention is observed. So these things leads to the diversity of proteins which are present. So due to the reason of this due to the reason, the lesser number of genes are sufficient to generate this diversity. So when we look back at the bacteria or some primitive organisms, some primitive eukaryotes for example, not the advanced eukaryotes, so they have limited number of proteins and limited number of genes. So if you talk about the bacteria, so they do not have introns, which are known coding regions. Their DNA is their DNA do not have those intronic regions, so that's why they do not have alternative splicing to undergo. That is why number of genes corresponds to number of proteins. But as we come to the eukaryotes, now they have intronic regions as well. So now the chances of increasing the diversity has increased in case of the eukaryotes. That's why we observe this diversity. So if we compare the different organisms, for example, I give you the example of eukaryotes starting from the C. elegans. So C. elegans, then let's move further to insects, the Drosophila, and then to fishes, zebrafish, to mouse, to human. So they have originated from a common ancestor. They have genes in common. But if you look at the proteins, then the diversity varies, because with the evolution, this machinery of spliceosomal machinery has improved, due to which more and more diversity is observed in the higher eukaryotes. And this, although this diversity has brought about the advantages for the complex eukaryotes, but in that case it has also increases the chances of disease. For example, the cancer. So why we are talking about these cancer diseases in case of humans? If we look at the mouse or some other organisms such as chimpanzee, monkeys, which are closer to human, so they do not have these cancers normally expressed. And even mouse also, they are not showing the cancer. Why? Because in human, as the diversity has increased, so the chances of mutations have also increased, due to which we are exposed to more of the disease as compared to them. And for the studies, when we select the models, so as we know that we cannot perform experiments on the live human because it is against ethics, so what people does is they perform experiment on the model organisms which are nearly similar to humans, such as mouse models or some monkey models or other models. So in that cases they have to induce these diseases. They have to provide such antigens or some other proteins which could lead to the cancer phenotype, because normally cancer is not observed in them. So one thing which has also, means if there is some advantage of one process, then there are some disadvantages also associated with it. So in that way, if we are able to recognize the changes which are happening or the diversity which this uh, means gene has led to, so we can track that what are the changes which are linked to the cancer, and we can target those genes specifically. So those proteins which are synthesized by those genes, and by targeting those proteins we could be able to affect, means we could able to control this disease. And in different people, so I'll discuss with you about the epigenetic modifications. So what happens is that if there are two twins and they are identical twins, and if one twin so one of them is living in India and another goes to America, So there will be change in the environmental conditions, different food items, and different things. So multiple environmental factors have changed, so they will influence his genetic, so his makeup. So epigenetic changes will come up. So his their genes are same, the genome makeup is same, but how these genes are expressed, they will differentiate because of the environmental exposure to the different environments they are exposed to. So due to which they will vary in their phenotype. So although they have same genotype, but they will vary in their phenotypes because they are influenced by the environmental factors. So environmental factors is also one of the important thing to be considered. So whenever we are designing the drug, why we talk about personalized medicine? Because different people have different expression profiles, different proteins are expressed at different extent. So we cannot design a single drug and say that this will cure both these patients. No, different people have different genetic and proteomic makeup, so we have to design the drugs which could be specific for them. That's why more and more approaches are going on, so that more and more countries come together and better personalized medicines could be designed, because the studies are mostly performed by the western nations. For example, America, Africa, other nations have performed the studies and they have designed different drugs. So it's not that their drugs will also help us, because we have to perform the study at our nation so that we could design the personalized medicine for our country, because our people are different, our environmental conditions are different, so our drugs will also be different. So that's why the personalized medicine is most important, and due to which people are working on that and why we need it. So okay, thank you so much. That's all about the session. Thank you so much for staying for this much time, because 2 hours are not less. So thank you. I am ending the session and I will see you the next week, Tuesday. Okay.